The overall goal of this procedure is to quantitatively detect protein-protein interactions in a transient expression system.
This method can help answer key questions in plant signal transduction field, such as how to quantitatively monitor protein-protein interactions after a chemical or environmental treatment. The main advantage of this technique is that it uses a luminometer or a CCD camera to detect the Luciferase activity, which represents the protein-protein interaction intensity, so the results are more accurate.
To begin this procedure, add 1 milliliter of a solution containing 5% sodium hypochlorite and 0.1% Triton X100 to a 1.5 milliliter microcentrifuge tube containing seeds. Let the solution rest for 5 minutes to sterilize the seeds. Then, wash the seeds with sterile water 5 times. Suspend the washed seeds in 200 microliters of sterile water. Using fine tips, carefully place individual seeds onto the surface of plated MS agar medium. Store the plates at 4 degrees Celsius for 3 days to synchronize germination. After this, keep medium plates under normal growth conditions for 10 days. Transfer the germinating seedlings into soil, making sure not to damage the roots. Cover the tray with a transparent plastic cover overnight to maintain humidity. Grow plants in a controlled growth room for 7 weeks, when they'll be ready for Agrobacterium tumefaciens infiltration.